Hello everyone. So today we are going to talk about cell, specifically about plant cell. So first we need to know what is a plant cell. The word cell, it is derived from a Latin word which means cella. It is also known as a small room. The cell is the structural and the functional unit of all known living organism. It is the smallest unit of an organism that is classified as living and is often called the building block of life. Some organisms such as mostly bacteria are unicellular and other organisms such as human, they are multicellular. Now we talk about further, cells are of two types. First is prokaryote and second is the eukaryote. The prokaryote word comes from a Greek word which means pro, before, and karyon which means nut. Prokaryote are organisms made up of cells that lack a cell nucleus or any membrane in case of organelle. Second is eukaryote. This word is also derived from a Greek word which means well or good, and karyon means nut or kernel. Eukaryotes are organisms made up of cells that possess a membrane-bound nucleus that holds genetic material as well as membrane-bound organelle. Further we will study about cell theory. So M.J. Skeldon and T. Szechuan put forward their cell theory in 1839. The cell is the structural and functional unit of all the plant and animal. Rudolf Virchow added 1858, this new cell arises from the division of pre-existing cell. The jelly-like substance in the cell was called as cytoplasm by Albert Collikus in 1843. Hugo von Moe in 1846 suggested that cytoplasm and nucleus together be called as protoplasm. The invention of electron microscope was done by E. Ruska in 1931. Now we will talk about plant cell. So the discovery of the cell was by Robert Hooke in 1665. He used cork slice to examine under a crude microscope. He could see only cell wall as the material was dead. Robert Brown in 1831, he observed a dense spherical body in the living cell which was subsequently termed as nucleus. Now we will further read about introduction to the cell. So plant cells are the basic structure and functional unit of plant. It play a crucial role in the growth, development and overall functioning of a plant organism. These cells exhibit a high degree of organization and specifications, allowing them to perform specific function essential for the plant survival. One of the best way to understand the intricacy of plant cell is by examining its cross section, which you can see in this diagram, which reveals the various structures and their function. So one by one we will read about all of them. The first one is cell wall. If we talk about the structure, the outermost layer, rigid and composed of cellulose. If we talk about function, it provides definite shape and rigidity to cell. Cell wall gives protection against pathogen along with mechanical support. It also prevents the osmotic bursting of the cell. The system of adjacent cell wall throughout the plant body constitute the apoplast. The plasmodesmata in cell wall form a system of interconnected protoplasm called symplast. Second one is cell membrane, also known as plasma membrane. If we talk about the structure, it have a thin flexible barrier surrounding the cell, and if we talk about the function, it regulates the passage of the substance in and out of the cell, maintaining internal balance. Third one is cytoplasm. If we talk about structure, it have got a gel-like substance filling the cell interior, and if we talk about the function, it have like houses organelles. It facilitates cellular activities and serve as a medium for various metabolic reaction. Fourth one is nucleus. In the you can see also the diagram of it. So it is present in all the eukaryote cell except for like mature sieve tube cell of plant and red blood cell of mammal. Nucleus is the central organ of a cell and the heart of the cell. It has a controlling influence on the heredity and activity of the cell, and it was discovered by the Robert Hooke in 1831. The diameter of the nucleus range from 5 to 10. if we talk about the function of nucleus, it contains the genetic information in its chromatin for reproduction, development, metabolism as well as behavior of organism. It controls all the cellular activities in the cell, and nucleus take part in the production of ribosome. Fifth one is endoplasmic reticulum, also known as ER. The term was coined by K.R. Potter and Thomson in 1947. The ER is the single unit membrane structure, 50 to 60 thickness, and is a network of tubules and form the vesicle system. ER occur in three forms. First is cisternae, then vacuoles, then tubules. These three forms may develop in a single cell at the same time or at different time during cell cycle. Cisternae are long and flattened unit, 40 to 50 mu thickness. Vesicles are round, 25 to 500 mu in diameter, and tubules are diverse in shape, 50 to 100 in diameter. Cisternae are commonly found in the cell and responsible for enzyme and protein synthesis. This is a diagram of endoplasmic reticulum. Then further, if we talk about the function of ER, it produces all the lipids, proteins and required for like synthesis of new cellular membrane. It can be like regarded as a membrane factory, and ER provide a space for the storage of synthesis production like glycogen. ER give mechanical support to cytoplasm as accessory or cytoskeleton. It increases surface area of the cytoplasm for the metabolic activities, and it play a role in the origin of plasmodesmata. Sixth one is the ribosomes. Ribosomes were first observed by Robinson and Brown in 1953 in plant cell and by Palade in the same year in animal cell. Ribosomes are also called plate granules, may associated with like ER or in cytoplasm in floating state. It is also found in mitochondria and chloroplast and are numerous in meristematic cells and less in lesser activity cells. They are undeveloped smallest cell organelle, and its size is from 150A to 230A, and they are of two kinds, 70S which is found in prokaryote cell and 80S which is found in eukaryote cell. Ribosomes found in mitochondria and chloroplast are similar to like 70S ribosomes. We talk about the function of the ribosomes. So ribosomes are the sites of protein synthesis. Ribosomes get attached to mRNA and form protein synthesizing apparatus. Ribosomes provide necessary enzyme and proteinaceous factors to catalyze the process of protein synthesis, protein synthesizing apparatus, and ribosomes protect mRNA strands against nucleus. Ribosomes also protect the nascent polypeptide chain against the action of digesting enzymes. Seventh one is Golgi apparatus, also known as Golgi body. If we talk about the structure, it have a stack of flattened membranes. These were discovered from the nerve cell of a barn owl by C. Golgi, 1891, and they are universally distributed in plant and animal cell except for bacteria, cyanobacteria and mature sieve tubes. The number of dichotomes per plant varies from like 1 to 25,000, and they are most abundant in meristematic and secretic cell. They are made up of like three main components, which is cisternae, tubules and vesicle. Now cisternae, they are the group of dense flattened parallel sacs of variable shapes and size which are stacked one above the other. You can see in the diagram. And vesicles are clustered of small spherical structure bound by unit membrane. They are found around the edge of cisternae and are derived from the edge. The vesicles are of two types. First is a smooth wall and sculptured, and the form contains secretory material, while the function of the latter is not known yet. If we talk about the function, many molecules, for example, scattered protein, glycoprotein, glycolipids protein or lysosome cell walls material are received by Golgi body, and then molecules are secreted through secretory vesicles into the cytoplasm. Lipids synthesized in CR are transported through Golgi apparatus to the plasma membrane, and Golgi bodies play an important role in the formation of lysosomes, yolk, melanin, and secretion of hormones. Golgi body synthesizes hemicellulose and pectin groups of polysaccharides during cell division. These substances are deposited on the cell plate. 8.1 is mitochondria. If we talk about the structure of mitochondria, they are double membrane structure made up of two unit membranes. Outer membrane is smooth and 68 to 75 a thickness. The inner membrane goes into fold called cisternae, and thickness of the inner membrane is 50 to 70 a. The space between the two membranes is called intermembrane space or perimitochondrial space or outer chamber. The inner membrane bears knob-like structure called oxysomes or elementary particles. The cavity of mitochondria is occupied by homogeneous dense fluid material called mitochondrial matrix. Matrix is the site of Krebs cycle, fatty acid, oxidant and amino acid synthesis. This is a diagram of it. Now we talk about the function of mitochondria. Major part of APT is synthesized in mitochondria by oxidation of foodstuff. It provides a raw material for synthesis of chlorophyll, cytochrome, primitives, alkaloids and steroids. Mitochondria is the site of amino acid synthesis and fatty acid synthesis. All the necessary enzymes for respiration are located in mitochondria. In prokaryotes, enzymes are respiration are located on the plasma membrane. 9th one is chloroplast in plant cells. So chloroplasts are green plastids found in green tissue of eukaryotic plant cell. The shape and size of chloroplast may be spherical, oval or disc shaped, spiral, spirogyra, stellate, zygemma and reticulate oidogonium. Distribution of it like chloroplasts are distributed throughout the cytoplasm of all green cell of plants except autotrophic prokaryotes. The chloroplast is bounded by two unit membrane which is made up of lipoprotein. An external surface of outer membrane is smooth and freely permeable to small molecules. The inner membrane of the chloroplast is selectively permeable as in mitochondria. The space between two membranes of the chloroplast is called the inner membrane space, and the inner membrane surrounds the ground substance which is colloidal in nature and called stroma. The lamellar system which are sac-green bodies are called grana. The term grana was coined by Mayer in 1883. This is the diagram of it. Then if we talk about the number of grana, it varies from 10 to 100 in each cytoplasm, and the granum consists of a stack of 10 or more thalloids arranged in a definite or order. The thalloids membrane connecting one grana with the other are called intergranal lamella or stroma lamella. Grana are not found in the chloroplast of algae and they possess lamellate chloroplast. Thalloid membrane are the site of light reaction of photosynthesis which produce this product of light reaction ATP and NADPH plus H plus diffuse into stroma from grana. The 10th one is vacuole. The structure is large membrane bound sac containing cell sap. The function of vacuole is that they maintain the turgor pressure which is important for the structural support and control of the water movement. They are storehouse of the reserve product and accumulation of the cellular metabolites by byproduct. Protecting substance and toxic substance occur in the vacuole. Thank you.